Assalamualaikum and hi everyone. Today I would like to share with you about our research paper, a nano green synthesis approach using plant-based material for antimicrobial agent. So, this is a part of Inventor that you can see. Okay, for the first slide, I will explain more about introduction. Silver nanoparticle AGNP have an interest in modern research and is known as one of the important nanostructure in developing science and technology application. Besides that, silver nanoparticle, or we call it as AGNP, are a novel material that is antimicrobial, biosensing, superconducting, and electronic material due to their unique characteristic. A consistent increase in global market demand of the industrial nanomaterial market due to its multi-purpose properties. Moreover, green nanoparticle synthesis is a new field of nanotechnology. Green synthesis has been proved superior to the physical and chemical methods since it is a cost-effective, environmental friendly, and easy scale-up of mass production. So, for the problem statement, there are three problem statements. The first one is an inefficient supply of silver nanoparticle in a various industry. Drawback from the physical and chemical method in producing silver nanoparticle. And the last one is the lack of research of metal reduction chemical from algae, particularly in AGNP. Next, research question. There are three types of research question. The first one, what are the environmental approach to synthesis to the silver nanoparticle? The second one, how to determine the morphological and chemical analysis of the silver nanoparticle. And the last one is the how to evaluate, is there any microbial activity of produced SI. For the research objective, the first one is to conduct a green synthesis approach by reducing silver nitrate to silver nanoparticle utilizing a sewage algae bloom extract. The second point, to characterize the synthesis nanoparticle by morphological and chemical analysis. And the last one is to evaluate the antimicrobial activity of biosynthesis nanoparticle. So for the project scope in this paper is this study to aims to assess the green synthesis strategy of AGNP by using a unique biomaterial sewage algae bloom extract, which is rich in nitrogen and phosphorus. Besides that, the characteristic of silver nanoparticle will be identified. UV visible spectrophotometric to analyze the formation of AGNP. The second one is SRD to show the size of the crystalline structure of AGNP. FTIR to examine the molecular configuration of AGNP. SEM, AFM, and TEM to identify the shape and size of AGNP. And the last one is HPM and 3D nanoprofiler to identify the surface structure of AGNP. And lastly, the antimicrobial activity will be further confirmed by identifying the zone of inhibition of the agar plate. So, we move to the literature review. Silver nanoparticle, or we call it AGNP. Initial silver ions and compounds were found to have a natural ability to treat health-related concerns, and they were also known as a hygiene compounds. AGNP with diverse structure and properties was great with a high anti-infection component of healing reason. In contrast to other types of nanoparticle, AGNP is non-toxic, leading to the production of health products. So, here is some overview of synthesis of silver nanoparticle. For the first one, there are two types of top-down method and bottom-up method that you can see. So we move to the biological method. Three components influence biological synthesis. The first one is dissolvent, reducing agents, and non-toxic substances. By using the natural resource material as solvent and reducing agent, the biological technique appears to provide the controlled particle size and shape, which is a critical element in a wide range of biomedical applications. So, biological method is simple, cost-effective, dependable, and environmental friendly method. So we move to the next slide. Algae. Algae are diverse group of unicellular and multicellular autotrophs. Algae biomass both live and that is employed in the biogenic production of nanoparticles. Live in both marine and freshwater habitat and terrestrial ecosystem that are damp. The rigid cell wall matrix contains a variety of functional groups. We move to the next slide. Sargassomaticum. Sargassomaticum or Japan's wireweed is a big brown seaweed native to Japan. This species have been utilized to synthesize various metal nanoparticles in environmental friendly manners. Sargassomaticum's extract have a high polyphenol contents and anti-cancer potential. Besides their therapeutic benefit of sargassum species include antiviral, anti-allergic, and anti-tumor.
Next is the methodology. For the flowchart, the objective one is green synthesis of silver nanoparticle. First, we will collection and preparation of the sewage alga bloom extract, and then synthesis of silver nanoparticle with a biological method. For the objective 2 is the categorization procedure, optical structure and a surface chemical analysis using the UV-Vis, SEM, FDR, XRD, HPM, and others. For the objective 3 is evaluation of antimicrobial activity, which is determination of the zone of inhibition. The synthesis of silver nanoparticle from the biological method and the alga extract will be evaluated based on the inhibition zones of the microbial activity on the alga plate. Next, this is the overview of the research. First is the preparation of the sewage alga bloom extracts, and then synthesis the silver nanoparticle using the optical structure and surface chemical analysis. Next is the evaluation of the antimicrobial activity. This is the methodology, which is first preparation of the sewage alga bloom extract and the synthesis of a silver nanoparticle. First, collect of the alga, and then dry the alga, fine powder alga, and then centrifuge, heatings, filtration, alga extracts, and stirrings. Next is the synthesized silver nanoparticle, and the last one is synthesized silver nanoparticle in the powder form. Next is the categorization of the analysis. This is the apparatus that we will use, which is UV visible spectrometers, 3D nanoprofiler, TEM, SEM, AFM, HPM, XRD, and FTIR. For the antimicrobial activity, first collect the sewage alga bloom and then dry the alga to be extract, and then the alga extract solution will be added with the alga silver nitrates, and then the silver nanoparticle synthesis when the colors of the silver nitrate turns dark browns, and then we will go to the inhibition zone. That is the antimicrobial activity. There is a two-step, which is a serial dilution method and the diffusion access method. For the serial dilution method is for the preparation of the sample. First, preparation of silver nanoparticle sample, and then preparation of E. coli and B. subtilis cultures. Next is deposition of silver nanoparticle sample of E. coli and B. subtilis culture. Next is the measure of the inhibition zone thickness. Next, we will go to this diffusion as a method.
Result and discussion. Visual identification of synthesis silver nanoparticle. The pale yellow color produced by the addition of silver alga bloom extract to a colorless silver nitrate solution. As for change in color to the dark brown, it took 24 hours of incubation for the change to become noticeable at room temperature. The product formed after 24 hours was in a valid form that settled down at the bottom of the centrifuge tube at 10,000 RPM. Characterization of silver nanoparticle. First, UV-Vis identify the development of silver nanoparticle. Wavelength obtained at 390 nanometer indicate a relatively small grain size and even distribution of the synthesis silver nanoparticle through the solution. Second, XRD reflect toward the crystallized size of silver nanoparticle. There are the table of the result of silver nanoparticle compound. Next, FTIR. In the photo, we can see the functional group that include in this silver nanoparticle. After that, 3D nanoprofiler. 3D nanoprofiler image 3D A, the agar extract surface was smooth and sharp as can see in the image. Specific data at the B, the height, area, size, and pitch the agar extract were visible noted on the table. C, the surface 3D nanoprofiler image 3D view, the silver nanoparticle surface was sharp, smooth, as can see in the image. Specific data B, the height, area, size, and pitch the silver nanoparticle were visible noted on the table C, surface, roughness, and waviness. Next, HPM and SEM. HPM, the agar extract was more smooth surface arrangement. Silver nanoparticle was produced with rough arrangement with the different irregular structure particle. SEM, image of irregular shape raw powder silver nanoparticle, triangle, pentagon, and hexagon shape with diameter up to 100 nanometer. After that, TEM and AFM. TEM, the size of the shape of silver nanoparticle was studied using the TEM. The TEM image revealed that the silver nanoparticle synthesis was spherically shaped particle with smooth edge. The size of the silver nanoparticle range between 10 to 100 nanometer with an average size of 50 nanometer. AFM is more precise in diameter with lateral size image that sample in 3 dimensions and allow the characterization of the nanoparticle height. Next, antimicrobial activity. Antimicrobial activity of the tested compound with for 1 ampicillin, 2 distilled water, 3 silver nanoparticle, and 4 agar extract. Again, B subtilis at the bottom, A, B, and C, and E. coli, D, E, F, with concentration of silver nanoparticle and agar extract 1 milligram, 0.5 milligram, and 0.25 milligram. Conclusion and recommendation and commercialization. All of the 3 objectives that were mainly emphasized on the green steel road were successful achievement. The elusive role of nanomaterial with small size can be produced, thus create a high prospect in utilizing small amount with more reactive directly to the target and expected to become more sustainable usage. Commercialization impact applicable for antimicrobial biosensing, superconducting, and electronic material. Cheaper and readily available resource with higher commercial value. The preparation method are not involving the hazardous material. This is the achievement. Next, this pattern filling, the preparation method for synthesis nanomaterial. This our poster presentation. Thank you.